Well, hello everyone, and welcome to another NAMA webinar. I'm Trent Blizzard, the president of NAMA, and I am here with Dr. Rick Vanderpool. Say hi.
Hey, everybody.
He is
All
going
right.
to present to us today on how to recognize and conserve rare fungi in your state or province, as the case may be. Um, and now, I think probably what we'll do is we'll get started here, Rick. But first, let me just kind of say a little bit about you for people that don't know you. I met you for the first time this year at the NAMA annual foray, and you were our keynote speaker, and just you killed it. People loved your presentation, and they loved your
Thank you.
energy. And you also led forays and were really involved. And NAM was really grateful for kind of all that you brought to our event. And for people that don't know you, you've been doing this for, I think, almost 50 years. You studied in San Francisco at Dr. Tears. You taught at several different places. I know Antioch was one of them. And now you live in New Hampshire and you're very involved in conservation. So I don't know that I don't really want to read your bio for everybody, but I would
It's fine.
encourage everybody to check them out or you can tell us more. And I think if you
Yeah.
want to fire up your slides, I'm
Yeah.
gonna bow out of here, and we're recording this. So for anybody that has to leave early, you can come back and check it out. Otherwise, I'll turn it over to you and say thank you.
Okay. Great. Thanks, Trent. Appreciate the time very much. And it's a pleasure to be here with everybody tonight, albeit remotely. Um, how we've learned to use technology to our advantage. What can I say? I'm old school. I remember slides getting stuck in the machine before they dropped into place. I remember the times when they got stuck and burned in place because the lamp was too hot. So yeah, I've been doing this for a while. But this is something that is near and dear to my heart, talking about rare fungi. I've been at this, as Trent said, for over 45 years. My first real hard class that I took in mycology was in 1978. And I've been sort of struggling with it ever since because I thought birds were pretty good when I was growing up. And then I learned botany, and I was a botanist, hired gun botanist for a while. But boy, when I hit the mushrooms, that just, it's kind of eating humble pie, as you all know. There are just hundreds and hundreds of thousands of species, and so much we don't know. But one of the things that struck me very early on, because I was involved with rare species conservation from the get-go, was the fact that very little had to do with fungi. And in fact, nobody really knew how common certain species were. All that I could ever hear was, well, let's see if we can find some oysters or chicken of the woods or hen of the woods or oh, what can we do with that hemlock varnish shelf? And it was all the common stuff. So in 1996, which was I believe the last joint NAMA NEMF foray, and NEMF, for those of you who don't know, is the Northeastern Mycological Federation that I've been a part of for 40 years. And NEMF and NAMA joined together in 1996 at Ascutney, Vermont. And my part in that sort of foray was, as co-chair, was to help oversee not just the forays, but also to invite faculty. And I had the ability to sort of underscore the title for the foray, and it was called Conserving Mycological Diversity. And I did so because I wanted to show people what folks in Europe were doing with conserving rare fungi. And the red list concept was pretty new. Not very many people in the United States had heard of it. Certainly some mycologists had, but the three faculty that we invited from Europe just opened all our eyes to what the possibilities were. A, that you could actually sort of wrap your hands around the diversity of fungi well enough and track it over time that eventually you get to know which ones were rare, which ones you didn't see very often, or better yet, as the red listing has pursued its objective, identifying those that are in decline, those that we used to see, say, in the 1900s or even the late 1800s, and those that had disappeared since that time. So that was a real eye opener, that 1996 foray. And since that time, I've been actively involved in trying to get people interested in not just recognizing fungi and the species that they have in their area, but looking at the ones that are rare. And of course, as all of you know, sort of the era of DNA came in and it just blew the lid off of everything because all these species that we thought we knew, right? Oh yeah, that's a Pluracibella porogens. Well, not really. It's Pluracibella SPN01 or some other variant that the genome sequencing has opened our eyes to and really showed us that we've got many, many more species out there than we thought. And some of these are, in fact, turning out to be quite rare. And through the combination of our, review of historic records and looking at some of the sequence data that's coming in, I think we're going to make some pretty good headway in the next couple decades in terms of conserving rare fungi. But first, we've got to learn how to do that. So that's what I'm going to spend a little bit of time talking about. I'll start with some very basic questions like what makes a fungus rare? Okay. And some of you are probably well aware of this in the plant world or the animal world. The edge of a natural range is a common phenomenon that produces some type of, distributional data that shows that something's not very common anymore as you go north, west, south, east. Also, there are some species that have very selective habitat conditions, especially the microhabitats. Certainly, there are moisture requirements. In the picture there, I'm holding what's called a tricholoma, um, and this is the grave or dense tricholoma. It only occurs under heavy rainfall conditions, and you have to have this sort of long period of sort of gestation for this to occur. And so in my region, we had basically two different fruitings in 40 years. And when it did fruit, there was, a fair amount of occurrences, but then it was another 20 years before it happened again. So you have these conditions that really drive the distribution data readily. And sometimes we understand them and other times we don't. For example, in some ectomycorrhizal or even endomycorrhizal species that we can attach to certain plants like Atlantic white cedar, yeah, that makes sense. We listed one of our Northeast Rare 20 species on that basis, actually a couple of them that are associated with white cedar swamps, and that's a declining resource. There are Saturnid moths that support cordyceps species, and some of them are very specific to those Saturnid moths, and yet we've lost a lot of those moths due to taconid flies that we introduced for control purposes. And I think our Saturnid population is less than 1/10 of what it was 100 years ago. And then, of course, you have these hosts that certain mushrooms like Squamanita rely upon. And how common are those hosts? It's very difficult to get a sequence on the host in a Squamanita. You can get it off the fruiting body, but it's difficult to really identify the species that Cicidium is growing on. And then last but not least, there are these evolutionary outliers. And those are ones that I find fascinating. Um, I was reading Bill Bryson's book, A History of Almost Everything, and reminding myself through this book that, we've lost over 99% of the species on this planet since the evolution of life began. And that's sort of a humbling reminder that some of our species are in fact going to be that way. They're going to disappear on us, and we really can't do anything about it for reasons that we either understand or we don't. So there's this sort of evolutionary piece that I wanted to introduce to you as well, and I'll get back to that in a minute. So, what are some of the threats, right? We have lots of threats to a variety of animals and plants. What are our threats to our fungi? And certainly, the loss of forested habitats is probably one of the greatest threats that we've got in terms of North American declines of macrofungi. And it's fairly evident that when you've got, especially with the mycorrhizal species, but even saprotrophs, and there in the middle picture, you're looking at a saprotroph that is tied to hemlock. Well, in the southern Appalachians, you don't have to look hard to find hemlock woolly adelgid otherwise known as HWA. And if you lose hemlock, you lose Ganoderma tsugi. And so that's a good example of a loss of a saprotroph that's attached to a tree that is being threatened by a pathogen. Another good example is right below it, and that's emerald ash borer that's pretty much taking out all the ash in the eastern US. I can't remember exactly where it started, somewhere in the middle Atlantic states, but it's well throughout all of the Northeast now in the middle Atlantic states. And I kind of chuckled when at the foray this September, we found a fair amount of this Boletonellus merulioides, which you see to the left of the beetle. And it's a pretty common bolete, right? It's under every ash tree. Oh, wait a minute. There is no more ash. So it's now sort of elevated itself because of that association into a species of conservation concern, at least in Vermont, and it also is in some other the other northern New England states. And then, of course, you've got fungal invasives. Many of you know probably bet more than I about how yellow oysters have taken off in the woods in the eastern United States and become a real sort of aggressive invasive by taking up good habitat that other species of saprotrophs would otherwise normally take up. And not too very commonly, but certainly in some situations, you've got sea level rise affecting fungi species. I thought this was kind of, yeah, really? How many saltwater fungi do we have? But there are a few, believe it or not. And Jean Berube from Quebec pointed one out to us. It's pretty rare back about 13 years ago. And I was like, huh, that's a salt marsh species. How about that? So there are some, but that's just another example of some of the threats that we see. So then why do we want to conserve them? Well, I mean, my first point is why not, right? I mean, we've got these great tracking lists for animals and plants, and why not? Fungi, in so many ways, are older, certainly, than most of our animals that are extant today, older than most of the plant species we see on the planet today. They also represent, one of the most essential links to the integrity of ecosystems. That's a given that we're only learning more and more about every day. And they've also developed these symbiotic relationships, like I said, mycorrhizal relationships, sometimes with rare organisms. And in fact, when you look at things like Atlantic white cedar, here in the East, you wouldn't have Atlantic white cedar without the associated species that are providing it with the nutrients, just the same way that you find these upland species doing, oaks and chestnuts and so forth. And then, of course, you can find in certain situations fungi that are representative of soil conditions and they act sort of like as a canary in the coal mine. We noticed that with the loss of certainly biodiversity in Europe. And that was one of the real drivers for the red listing to see how many species of fungi were lost due to acid rain back in the 50s and 60s. And that really, they cut their teeth on understanding how fungi can disappear when things like that take place on a global macrocycle basis. And of course, they're also very reflective of specific, environmental conditions that we need to pay attention to. I pointed out the ones associated with those with emerald ash borer and hemlock oleodelgid. And at that level, when we're changing stands, stand level alterations, fungi are going to take a hit, and they're also going to have cause a destabilization of those ecosystems that are forested. So we really need to pay more attention to them. So how do we begin? It's a big job, and we've been at this for some time. And I say we because several of you are on this webinar have helped me along and have been doing this for quite a while. And you know what I'm talking about. There's a lot of people that are doing this, but we need more and more folks to really engage this and get this, out into the public in a much bigger way. So the first thing that, and I'll use Vermont as an example. because I currently sit on the steering committee of the Vermont Fungal Scientific Advisory Group. And as I'll point out in a minute, Vermont's sort of taken the lead in New England in terms of adding fungi to their lists of conservation concern species. And so in Vermont, our the first question was, gee, what do we know about our state and province in this case? How many species do we have? Do we have an actual baseline list? So we started to mine the data. We actually wrote a grant and got a grant to have the Vermont Center for Ecosystem Studies download all the GBIF data, check for the catalog of life, COL, and then also look at iNaturalist records and mushroom observer records for the state. And that really got us to a good starting point. So that's necessary. You really got to, you know, considering that we're talking about, in this case, political boundaries for your political boundary of interest, you need to start with some baseline data. Then you really need to know what conservation measures are already in place for the other species, non-fungi species. And pretty much every state in our country, in the US, and all the provinces have listed species. Um, I did a fairly extensive search, as I'll demonstrate in a minute, for North American lists for rare things, and fungi are almost absent. So at least we can use or start with the models that we have, the state heritage program lists of endangered, threatened, and special concern species, the nature serve list. I'll get into some of this in a minute, but that's a good second place to go or to start with, and that's looking at your own state list. Then see if you can research what has already been learned about rare fungi. And it can be as simple as going into MycoBank and looking at all the mushroom records for a certain group over the course of 150 or more years and seeing what data you've got for your state. And if you notice trends, gee, we used to collect this mushroom regularly and we haven't seen it in 50 years or 60 years, then dig a little deeper and see if you can find out or try and posit some hypotheses as to why that might be, right? And of course, it's a daunting task when you consider the diversity of fungi, but very often popular ones are ones that are well known. And certainly some of the rare 20 that we crafted for the Northeast are very well known mushrooms. Certainly some of those you'll be able to find some information about. At least get a toehold and get a start on your list. And then last but not least, but something that you really need to do is, look at the threats, right? If you're going to use, for example, the IUCN Red List, and I'll talk more about that in a minute, is a way of, elevating information about rare fungi. You have to address the threat factor. You have to see if you can identify what the threats are. Oh yeah, okay, we've got hemlock woolly gelotrid taking out the hemlocks, and here's a mushroom that only grows with hemlocks. No problem. There's your threat, clearly identified. Okay, so that's another step that you need to take. And then, of course, last but not least, you want to try and get a starter list circulated with other folks, and in that sense, you really want to, well, yeah, get help. What can I say? You need to get help. So there are a lot of places where you can get that help, right? You've got supporting organizations across North America. So many of you are very familiar with Fundus, and we have now 4 rare fungi challenges. I'll talk a little bit more about that in a minute. We've got regional clubs and federations here in the Northeast, the Northeastern Mycological Federation of Quebec. You've got the Fédération Québécoise, the Groupe des Mycologues. Pardon my accent, I butchered it, I'm sure. You've also got this associations of fish and wildlife agencies. We have one in the Northeast, one in the East or Southeast, and they have regional committees for what they call regional species of greatest conservation need. That is that RSGCN. Okay, that's a pretty common term used in talking about anything rare. So species of greatest conservation need. And there are regional committees that support and research and update lists of all taxa. And here's another place where fungi are fairly poorly represented, but they're starting to make a toehold in the lichenized groups. You also have the USGS, which has its Science Analytics and Synthesis program, and they're the sort of national repository in the US for SGCN species. So they'll aggregate all the state lists together, and you can actually go to the SAS program and download lists for your state and see if there are any fungi on there or not. And mostly there won't be. And then there's also state natural heritage programs. And of course, they're the body that's generally designated in the state government to track rare stuff, right? So you've got in New Hampshire, for example, we have the Non-Game and Endangered Species Program, and they track all the animals. In our state, the Natural Heritage Program pretty much deals with plants. So then you've got some states that have both under one roof. And I know Hannah Huber is on this webinar, and Hannah is working in Pennsylvania at the Natural Heritage Program as a conservation mycologist. I mean, that just, when I first heard that, I was like, yes, we're making progress. We actually have somebody in a heritage program who's a mycologist and hired to do that. So this is the kind of sort of support that we need, and that's the kind of help that'll really help us get off the ground with making inroads into conserving fungi. And of course, last but not least, where would we be without our wonderful host organization, NAMA? Yeah, a lot of faces there. You may recognize people behind the scenes that have been doing this for a long time and are really contributing vast amounts of knowledge and talent to allowing us to make better decisions about the conservation of fungi. So when we look at the map of North America, you realize, yeah, it's a big job to try and wrap your head and hands around the diversity of biomes. In this case, you're looking at an ecological region map. There are 15 different regions. And as many of you know, within each of those regions, they're broken apart in different subsections and sections, and then ecotypes. And depending upon which sort of reference you use, the goal is to, for your region, your political boundaries, to break it down into the diversity of habitats where fungi can occur, right? So that's going to really help you out. For example, if you're down in the Southern Appalachians and the threat is hemlock woolly adelgid, you'll want to identify those habitats where hemlock is a dominant or co-dominant species in the stand, right? So that would be a good target place, let's say, to start researching what the records have been of fungi that you found there, especially those that are associated with hemlock. In Vermont, we've got through the FSAG group, we're trying to look for places where we don't yet have beech leaf disease and setting up baseline fungi plots, mostly doing macrofungi work, but sometimes eDNA as well, and then comparing that with sites that are otherwise similar but have now beech leaf disease that's decimating the population of most of the stand. And those kinds of comparisons are going to really help us out as well. But when you look at North America and then look at the number of records that are in GBIF for fungi, yeah, it's a daunting task. There's over 6.9 million observations of fungi in North America on GBIF. And so, and a lot of times, we've estimated fungi, as many of you know, between 2.2 and 3.7 million species. That could be a very low number. And yet we only have about 171,000 recognized in the catalog of life. So, we've got a lot of work to do, and we're going to be finding new stuff all the time. So the big question really at the end of the day of looking at the different habitats you're in is which ones are rare or possibly declining. And that's, like I said, not necessarily an easy task, but you can find some guidance in certain very common databases that we often very typically start with. For example, are they under an Endangered Species Act? In the US, we only have three species that are listed under the Endangered Species Act. And that in and of itself is kind of unique because the court litigation that occurred in the Pacific Northwest over rare fungi species, they came to the conclusion that, yeah, the ESA only specified animals and plants, but not fungi. And so for all the hard work that went into the National Forest Plan under the Survey and Manage guidelines, where there were 234 non-lichenized fungi and 81 lichens identified as rare in this northwest area of the US, they couldn't effectively use those guidelines and ascertain them as, species of conservation concern that would have an effect on, say, a logging timber plan or something like that. So, it's a good starter, but it won't be hard to figure out how many endangered species we have using the federal legislation. But even better and more current is the IUCN Red List, which I mentioned before. And I've got a number of sort of references that I'll go through for the next few minutes to point out where to look for rare stuff, rare fungi that we already know have information about. So you'll see there's about 1,100 or so species of macrofungi. And if you add in cup fungi, truffles and allies and lichens, there's what's listed so far for North America. Under NatureServe, you don't have any fungi yet. I understand they're working on that, but they've got 426 lichens that are listed in these 163 of which are under the G1. That's the rarest global rank you can get, G2 and then G3, plus a few historic species as well. In Canada, you've got what's known as the Committee on the Status of Endangered Wildlife in Canada. And they look at all taxa groups as well, except fungi. The only ones they include are lichens, and there are 23 species on their list of endangered or special concern lichens. I mentioned the Survey and Manage guidelines in the Pacific Northwest, which is again arose out of the National Forest uh, efforts to better understand what rare species we have. That was, by the way, the predecessor to why we even have two lichens listed under the ESA. That happened within a year after they finished the Survey and Manage guidelines. And then, of course, Fundis, we mentioned they have rare species challenges. We'll take a look at that briefly. And then state lists. Some states, these 7 right here, have their own lists of rare fungi. Most of them are lichenized fungi, but nonetheless fungi. And then last but not least, what they call the SWAPs or State Wildlife Action Plans. So Global Fungal Red List. This is easy. You just Google that and you'll pop in. Greg Mueller from Chicago Botanical is the lead organizer and manager of the Fungal Red List for the macrofungi. And that's something that he engaged me and a number of other people in 2012 to join a group of mycologists in Oregon to sort of kickstart the fungal red list for North America. There were hundreds listed from Europe, but really very few, if any, as I recall, from North America. Well, that's changed, as I indicated before. Anybody can nominate a species, and then the assessors are assigned to try and provide more information under step 2, do the assessment. That gets reviewed by the review committee for that particular taxa group. And then if approved, it goes on to the red list under one of their categories, which I'll summarize in a minute. Their totals, as they are shown by continent and fungal group, are as follows. And again, you'll see here in North America a total of 1,145 species, and 39 of those are micros, then the rust and smut group and the chytrids and so forth, the rest of the macros. So we're making progress. Again, this number was down, I think there might have been a 10 or 12 when I first approached this in 2012 under Greg Mueller's and Anders Dahlberg's tutelage. So we're gaining progress, and you can look at a lot of these species. You can look up each one, look at the distribution, you can read about the assessment of it, and you'll get good information about the threats that are have been identified for the different taxa. And if those taxa occur in your political region, then you can use this information wisely. NatureServe has, like I said, uh, all of the taxa groups except fungi as yet. And please correct me if I'm wrong, but I couldn't find anything on their website over the last couple of weeks. But here is a map that shows how they depict, in this case, endangered, threatened, and special concern species, as well as this one group called data deficient, or we just call them DDs. And those four groups are represented in North America, as you can see here. So if you're up in Alaska, wow, yeah, we got a lot of opportunity, I'll put it that way. Whereas you can see here in the eastern and the northeast in US, we've got a fair amount of data that's been fed into NatureServe. And they use status ranks, as I indicated before. On that map, we had these three groups as well as the data deficient ones. And this is I'll come back to this at the very last slide, but this is something that I think is of concern. Whether you can adequately identify a particular mushroom as being a GN1S1 or a G2N2S2, etc. That's kind of something that is going to be a bit of a challenge until we get a lot more data in. In Canada, like I said, they look at all of the different taxa groups except fungi, but they have some lichens in there, and they've got 5 of them on endangered, 9 threatened, and 9 special concern. And when you look at that total list of 880 species, it's really not that much when you think about the number of species of critters, especially in the arthropod group in Canada. So this is, how shall I say, a beginning. It's a start. It's better than nothing, but it's only a start. And then, as I said, Fundis has their rare fungi challenges. The West Coast was the first out the door. We launched that in 2020, and Elsa's on this webinar. She's one of the purveyors of not only, I think, this picture, but a lot of the information. Certainly, Biddy Roy and Noah Siegel, Christian Schwartz. There's been a bunch of people that have been really, really hard at work making sure the West Coast Rare Fungi Challenge has gotten off the ground. I started the Northeast Challenge based on data that I had been aggregating over the decades and proposed it in 2021. We opened it in 2022, and we took in 20 species to start, and we've done right well. I'll show you a map here in a minute of how many species and how many observations we've gotten. The Southeast Rare Fungi launched in 2023, the following year, and then the Rocky Mountains just got going this past spring. So we've got these 4 challenges. Each challenge has its own coordinator, and you can get the coordinators' names on the website, the Fundis website. Hannah's on this call, and I'm sorry I didn't see if anybody else is on this call to shout out to them, but you can see the regions that are covered by those 4 rare fungi challenges. And hint, hint, you guys who are in the Midwest, Trent, yeah, we need a Midwest challenge. It's probably already in the works. So as an example, the Northeast, like I said, started in 2022. We've had 14 of the 20 species observed. with entoloma, or some people call it Caloderma indigoferum, being the most often observed one, especially in 2024. A fair amount of records came in. Clavulinopsis appalachiensis, that was just a mind blower because we didn't have that in the first year, and there were very few records, and all of a sudden, boom, it was a great couple of years, and we've added a lot of good records to this species. And I think in some states it's, if at best it'll be a special concern, it won't ever get to a threatened or endangered category because there's just so many of them. And once we started looking, we found them. But then you get down to things like Tricholoma apium. We found one specimen. Thank you very much. You know who you are in the Northeast Mycological Foray in Cape Cod in 2024. And that was it. We've had only one specimen of Squaminidium bacchii. And there are 6 species, like I mentioned, that haven't even been found yet. So they're in the Northeast, I'll put it that way. Some of them have come in elsewhere in the country or up in Canada, but not in the Northeast. So these are the species that have come in, and as Hannah did this slide, she says, yeah, some of them have sort of won the case, and Entoloma indigoferum, as I said, Clavulinopsis appalachiensis, but not all of them have been found. And I mentioned I'd show you a map. This is a map of the species as of, I think it was last September when I finished this one. And just to highlight the records that we've observed since the Fundis Challenge started, they're all highlighted here. So here again, you've got a fair number of records, and most of these have come in recently. So it just points out how something like iNaturalist or Mushroom Observer can be really, really helpful once you start uploading layperson observations of species and then have them checked because they're in a challenge, how much more data you can get in a fairly short period of time. And also, if you, this will be part of the record, NAMA record for the webinar, you can scan the QR code and get a better look at which species were found where relative to the Northeast. So that's just the Northeast example. So the last thing I wanted to cover was looking at, in terms of source information, what states have added rare fungi to their wildlife action plans. And admittedly, it is a short list. This is really what I was pointing out in 1996, and I'll just keep hammering away at it. We literally have 6 states, with one, my humble state, because I insisted, not tracking them, but only looking, not putting them in the wildlife action plan, but tracking them in biotics. And when you look at most of what's being tracked, it's lichens. And the only real outlier is Vermont. And thanks to the Vermont Fungal Scientific Advisory Group folks, my hat's off to them. We put it together, and in the Northeast, Vermont is leading the charge. Why it has taken this many decades after having rare status conferred species in animals and plants and, algae and plant. I can't tell you how many states added algae, and yet fungi are not on these state wildlife action plans. And yet 2025, some of you probably know, it's a 10-year update. So the next opportunity to get fungi included in the SWAP will be 2035. So I'm hoping in another 10 years, we can at least double, if not triple, this list of states that include them, because it paves the way for people not only recognize the importance of fungi, but also integrating them into a whole suite of environmental assessments. And I just have this one picture here as a side note. I collected this last November thinking it was a squamanita. And it looked like it had a mycocystidium there. It had a separate cap that was coming up from it. I thought for sure it had something really Cool. I thought it was a squamata. Well, I was wrong. And thank you, Hart, for sequencing this. It came back as Phlegmasium triumphans. And I thought, okay, it's a Phlegmasium. So what? Well, guess what? There's only five records of this species in North America. So all right, so I didn't get squamanita, but I got another rare species, and I wouldn't have known that had I not picked it up and spent the time to dry it and send material off to get it sequenced. And my point here is simply that there's a ton that we don't know, and the more that we do in terms of collecting, vouchering, sequencing, and then accessioning these things in GenBank and in various microdatabases, the more quickly we'll be able to realize that long-term goal of conserving rare fungi. Here's another view of the northeastern states and how many states have animals and plants, and or in this case, vascular plants, excuse me, non-vascular, but fungi, again, a paltry small number. This is probably a little out of date. This publication was in 2024, but my point, I think, is well taken here. Vermont, like I said, once we had Vermont Center for Ecosystem Studies upload all the records, look at how many observations we had for fungi in Vermont. 59,000 records. And so that was a good start. That fulfilled that step one, get a baseline for what diversity you've got in your political region. And now the job, of course, is to tease that apart and find the rare stuff. Pennsylvania, Hannah just finished her listing of Pennsylvania fungi. I think it's up over 3,000 species, so they're well on their way for their first step in getting a baseline data set. And as I said, Vermont is already taken that jump into identifying rare species, tracking 34 of them, and now we're at the stage, which I'll point here in my last slide, in terms of getting to, the next sort of step. So once you develop the baseline, work with your local clubs. Contact your nearest lab tech facility that can do sequencing. Fundis is helping that out. Steve Russell, I think, is on this call, and his MycoMap project is opening the doors for a lot of us to send material to him. So that's another great resource. Assess the threats, like I said, for species occurrences. Check all the databases that already list rare species so you can get a handle on which ones you may want to add. Participate in a rare fungi challenge, see how you can challenge your own clubs to do this. And then we'll start working with natural heritage programs and see if you can integrate and try and inspire them to include fungi in their list because they can do a lot even before the next wildlife action plan is updated in 10 years. And in terms of getting support, there's a lot of different places you can do so, but I'll leave you with this thought that once you look at your species and you better understand your natural communities and the ecological landscapes that you're dealing with, before long, you'll get a lot better at least identifying where your assessment priorities are. So in that SGCN lists in every state, there's one list of stuff that we know about, and then there's a list we don't know enough about. The DDs, the data deficients, and these are your assessment priorities, and that's where we really need to shoulder our burden for the next step in conserving rare fungi. And with that, I'll just remind you, when you're out looking for rare fungi, you should always keep an eye out over your shoulder. You never know what you're going to find. Thank you very much. I'll stop sharing.
Thanks, Rick. That was awesome. Uh,
Cool. All
and
right.
totally entertaining and important. I saw a lot of comments coming through. I think people really enjoyed your presentation and all the information you shared. So thank you for that.
You bet. Oh,
Yeah, we have a lot of questions in here.
All
Do
right.
you want to read them in the Q&A and answer them, or do you want me to read them back to you? It's whatever you
Yeah,
prefer.
I can take a look at the
Great.
um boy. This one I won't be able to answer. Difference of opinion regarding dehydrator temperature settings. Who's done a Sanger sequencing for Rod Tullis and Brandon Matheny recommends 125. So that's maybe not my point because I'm not a sequencer. Um, yeah. So this one I can stab at. Are you working more with NatureServe ranking system or the IUCN Red List categories? They're really close to each other. When you look at vulnerable versus threatened and endangered, or what they call critically imperiled in the IUCN, it's very, very similar. So that's a really good question, and that's what the Vermont group we're actually going to have a sort of a little Zoom session next week about this, talking about ranking. How do you rank fungi? Do you do S1, S2, S3s or G1, G2, G3s? Are you doing the standard or are you using a different basis? I like the IUCN approach because what you're really required to do to get an assessment completed is to identify the threats. You identify the vulnerability, you identify the threats, and you have to identify the trends. And that takes some time. Some stuff, some species, right, takes decades to really get a sense of whether there's a trend. Yeah, okay, if it's Calabolitas peckii and it's a mycorrhizal with chestnut, guess what? We lost all our chestnut in the east, so that's not a hard threat to identify, nor a hard trend to identify at the same time. But I'll say one more thing, and that is that I came across a paper this week that sort of went, aha, this is a good concept. It was about orchid species and the plasticity of certain platantheras. And they identified a term which I hadn't heard before. It was called evolutionarily significant unit, or ESU. And the ESU concept, I think, and Gary Linkoff and I had many a conversation about this, gets us beyond the biological and gets us closer to the functional, to the ecosystem functional definition of what a particular species is doing as a mediator or a part of the stability of that system. And if we can move into a concept of that kind of level of understanding, I think we can get closer to a conservation principle, and a conservation policy, when implemented, will really make more sense on the ground than if we're just counting numbers of carpivores. All right, let me see if I can get to that. What's the salt marsh species? Oh boy, it's a Pseudomoruleus. Yeah, and I can't remember the species epithet. Jean Bruby or Annabelle Langlois will be able to help you out there. Uh, okay, rare seems to be an elusive term. Yeah, often now named via numerical codes to be considered rare. So here again, as I mentioned before with the sequencing, the rise in sequencing last 20 years, 25 years of getting a handle on species identification relative to a genotype. I think that's important, but I think if we waited until we got all those genotypes identified, we would have lost a lot of species of fungi on the landscape. So I'm not as concerned about Rashula NA01 or NB01 or PA01, whatever it is. I'm not as concerned about getting a name on it as I am about whether or not that taxon can be associated with a functionality in the system, right? And so that's where, we almost need to be doing that at the same time as we are trying to, uh, you know, publish a taxonomic name for something. Do you have a protocol for determining accuracy of INET observations? Wow. Okay, so really good question. And yeah, there's a lot of RG taxa out there that are misidentified. We know that. So here again, um, if there is a possibility, and certainly under all the rare species challenges through Fundis, we do this. If we get an observation in, the coordinator or somebody, a volunteer, will respond to that observer and say, "Hey, please get this vouchered and sequenced so we can confirm it." And that's really the approach that I would use with any rare species. I'd really suggest that you get it sequenced. And even if it's, 98.9% similar, because you don't always get 100%, as you all know. Um, and Rod Tula certainly schooled me up on how variable some of those genotypes can be as well, yet the morphology is exactly the same. You can get a little bit closer to what at least an identity is for a taxon and then move ahead. Midwest Challenge. Yeah, here, let's go. I want to see it. Um, is there any state in the US that has a fungal emblem? Well, there are several states that have state fungi, right? The mushroom, I should say, state mushrooms. Vermont notably passed a bill last year, or was it 24, um, uh, for lion's mane being the state mushroom of Vermont. And that's a cool way to do it too. That was, I think, one of the state reps for fourth grade class, did that. It was a great thing. And it just elevates that presence of mushrooms in the eyes of legislators and hopefully the people that make decisions about land use. In Quebec, we have official symbols for plants, trees, animals, and insects, but still no fungi. Could be a powerful way to trigger official recognition. I agree. And yeah, I agree. Where do you send your samples for identification? Um, right now, like I said before, I've used two different sources, as Trent indicated. Um, there's a there's a whole DNA sequencing committee under NAMA, and Hart's been accepting specimens on a limited basis because, of course, whatever the collection number at the annual foray was, but he's still getting through that material. And I mentioned Stephen Russell, Mycomap, is opening up projects in different states. So there's another possible source. I have a colleague of mine at UNH who does sequencing. So a lot of the universities usually has, Kathy Hodges, a number of people have capabilities for doing that. When a species is newly sequenced, is it automatically considered rare? And no, is the simple answer. Um, how much, I mean, look in GenBank, look at how many times have we sequenced, say, Amanita, Muscaria variegasaui, or whatever you want to, Amira muscaria, whatever it is, and how many times, I love Boletus edulis as an example, Boletus edulis, how many different types of sequences we've got in that complex. And here again, it just reminds me that, identification is one thing, but moving to placing it on a rare species list really requires those other three components I mentioned: threat assessment, its trends, and what its functionality is in a particular system, right? So that's something that I think is a requirement. Okay, another question here. How do folks working with other groups, plants, animals, etc., handle newly described species? Are there cases of recently described species that ended up on conservation lists? Seems like by definition won't have very much history and know much about precise ecological function. Good question, Nate. That does happen, and one of the best examples I think has been like birds. I used to work with a guy by the name of John Atwood. He sort of helped name the California blue-gray gnatcatcher. That came out at about, it was a rare species right up because it got identified as a particular, at first it was a variety, I believe, or a subspecies. I think it was subspecies. And so in the bird world, where you hardly ever get to name a new species because it just doesn't happen that often, that was in sort of instant fame, right? And I think the same is true for tropical amphibians. We've got some good examples of where certain frog species, tree frog species, have been named because they've been segregated out genetically, and to point out that they're, geographic isolates, for example, in the cloud forest of Venezuela. Um, there are a few other examples. Mammals, of course, I think, what do we get? One new mammal every 10 years or something. So it's pretty rare for that to happen. And other folks might be able to give you more specific answer to that question, Nate. Good question, though. George Christie, Rhode Island, we would not automatically place one on a rare species list. Good. We've had a rare species with two subspecies, and when those were elevated species level, both were obviously rare. We did that in Vermont with Lactarius cinereus variety fagatorum. That's the Lactarius that associates with beech, right? So that's why we listed that variety on our list of 34 because it's the one that's associated with beech. And I think it'll hold up. I think it has held up genetically. Um, there are examples when varieties or subspecies become important because they have that ecological identity, and that's the critical piece. Gordon asked, do you have any suggestions on how to approach the messaging around talking about the rarity mushrooms as a function of endangered habitat rather than something caused by humans over harvesting? Um, Yeah, if I understand you well, Gordon, that's a good it's a good reminder, you know. Um, Mushrooms, as you know, are fruiting bodies, and this is where there's a gentleman that I started to work with through the Vermont group who's doing eDNA surveys. And one of the things about eDNA, of course, is that you're optimally getting more information about perennial presence of a particular species in a particular habitat. And that can really help out when you're dealing with something like Tricholum agrave, which, like I said, only fruits heavily in 20 years, once in 20 years or more. And so you've got these species that may actually be fairly common, but fruit very rarely. And then you've got other ones that fruit on like abundantly. Sarcosoma is one of them that I can think of, and rich music northern hardwoods. And but that's where it fruits. That's the only habitat it fruits in pretty much. So you've got that whole range, right, of really uncommon species because they don't fruit very often. And I think one of our rare 20, the Hodophilus peckianus, is like that. And all the way to those that fruit all the time, but only fruit in one particular area. And I think we have to really be able to span, both ends of that spectrum in order to inform our rarity listings accordingly. And this is the last question, I think. I think that was it, Trent, unless I missed something.
Yep, I think that was it. So,
Good, good. Just about nine
yeah,
o'clock. That's awesome.
yeah, good timing. Big thank you again. This is a good chance for all of you that are still here to say thank you and sign off. And we will have this up on our website and YouTube here in a couple days. So thank you again, Rick. We're so glad you came and gave such a wonderful presentation,
You're
and
welcome, Trent. I
we're very
loved it. It's
grateful.
great.
Thank you. Good night, everybody.
Everybody, good night.
Bye bye.